Hello again, everyone, from the metabolic engineering class. This is the second half of the lecture that was supposed to be delivered about 2 hours ago. It's 3 o'clock now. Again, I apologize for my computer dying like that. Live and learn, as they say. So we were talking about metabolic flux, and we were looking at a metabolic pathway, or a fake metabolic pathway here, going from A to B. We have all our different enzymatic activities that'll take us there, and that is basically carbon traveling from reactant or carbon source, our A, to our product, B. And the velocities, the reaction velocities, the enzymatic reactions, all the sum total of all these rates, this will give us the flux, the J, which is the flux of carbon from one place to another. And basically flux becomes important when we're talking about steady state. So steady state will be reached when our intermediate metabolites, which are the spaces in between these arrows here, they will have adjusted so that concentrations, so that all the rates are equal, so that V1 equals V2 equals V3, etc., all the way to VL here. And again, we can think of that waterfall analogy that I showed you earlier, where we have all our pools that are going to be the size that they're going to be, which means that you've got carbon coming in and carbon going out at the same rate. So the pool's not getting any bigger and it's not getting any smaller. So essentially what that means is that we can easily break these things up into fluxes going from one node to another. And a node is anything like A, B, or C, or even this I. But what happens when we have a bunch of competing fluxes, let's say, or a bunch of flux that goes off in two different directions, like in this intermediate I here? Well, when pathways split at a given node, or intermediate in this case, our I here, there are multiple fluxes to consider. So you've got J1, which is our flux from A to I, but then you also have J2 from I to B, and J3 is our flux from I to C. And these fluxes can be thought of in such a way that you have J1, which is going to be equal to the sum of J2 and J3. And you may ask yourself, how is that possible? And again, this is all occurring at steady state, but how can this possibly be additive, you may wonder? This is 1, and then this is 1, and then this is 1. Well, how that doesn't add up? Well, these are J2 and J3 are not going to be 1, and this is why. So we look at this another way. We're basically thinking in terms of carbon, or moles of carbon. So if we have a split in the pathway, again, we have J1, we have J2 and J3, and J1 is equal to J2 plus J3, but we can think of it as two different pathways. The A to B pathway and the A to C pathway. They just happen to overlap from A to I here. They're sharing intermediates. So instead, this is J3. From A all the way to C is J3. From A all the way to B is J2. So we don't think of it necessarily as a split. We think of it as two separate pathways, even though these two arrows are the same enzymatic activity. But essentially what this is, if you have a certain mole value of A, say you have 2 moles of A, it gives you 2 moles of I. But then you're also splitting that in 2 moles of B and 2 moles of C. So you're not gaining on moles, because we can't create matter in this case. But we are looking at different types of products here. So this thing is splitting because, say you're taking a 6 carbon intermediate, turning it into 2 3 carbon intermediates. Well, the overall flux of moles of carbon, or moles, is going to be the same. You're going to have 3 moles of carbon here, 3 moles of carbon here, and here's your 6 moles of carbon going from A to I. So J1 is 6 C moles to I, and this is 3 C moles, and this is 3 C moles, so 3 plus 3 equals 6. And this happens a lot in metabolism, where we have a bunch of pathways and they branch off at certain locations, at certain intermediates, and those are called key nodes. And we'll talk a lot about metabolic nodes in the class. So in this example, we have A, which can make B, C, D, E, and F, and it's branching off at different intermediates. In these I1 right here, where A to B is, I2 is where A to C and or D is, and I3 where A to E and F are. These are different intermediates, and these are different nodes in the pathway. And each pathway, which is represented by its own arrow, is going to have its own flux. And from A to I1 is going to be one flux, whereas A to all these other intermediates, or all these other products really, B, C, D, E, and F, the sum of the fluxes to those are going to be equal to the flux A to I1. And so there are key elements of understanding pathway flux. There are key elements of pathway flux in general. So, first, let's define a pathway. What is a pathway? A pathway is a sequence of feasible and observable reaction steps that connect a specified set of input and output metabolites. It's what we've just been talking about, our A to B, or our A to C, or A to D, those types of things. Those are pathways. And the flux, the pathway flux, is the rate. It's a rate at which input metabolites form output metabolites. Feasibility, I talked about this at the beginning of the lecture at 1 o'clock. It's self-explanatory. Can these reactions be done? Are they thermodynamically possible? If they're not possible, well, then they're not going to be a component of the pathway flux, because they're not going to be a component of any pathway. The reaction must exist. The enzyme also must be present in the cell. If you're adding an enzyme as part of your metabolic engineering effort, adding a gene that makes an enzyme to make a unique product, then that you're introducing some feasibility to your new pathway here in your specific organism of choice. Observability, well, if we can't observe a reaction sequence, then it is of no value, because there's no way we can quantitate it. So we have to be able to observe the value of A to I, or what have you, some reactant to some intermediate or product. We have to be able to observe that. Once we can observe that, we can give it a flux and a rate and all that. So we're just kind of doing our metabolic bookkeeping, making sure we can keep track of where the carbon goes. So if we can't keep track of the carbon from each enzyme to each enzyme, then we can just group everything until we can observe and make sure that we have, all our moles of carbon accounted for. So one of the things we do is we examine and manipulate factors that are responsible for controlling metabolic flux, because you want to perturb the system to see, well, what do I get? Do I get more product? Do I get less product? Do I get more byproducts? Do I get different byproducts? So there are 3 steps to doing this. So first, we can develop the means to observe these pathways and fluxes. So quantity, or moreover quantitation, is the key here. So I can put in a certain amount of carbon source, and then I'm going to measure what I get out. What are the quantities of the things that I get out? It goes back to that think, pair, share, where I talked about a snapshot of the metabolites in a given cell under given conditions at a given time. So we can get a lot of these snapshots and get an idea of quantities that are flowing through certain pathways. Then, once we know this, or have this pretty well established, we can introduce perturbations to our reaction network. We have a certain reaction network that we're interested in. Perhaps we're interested in glycolysis and producing some kind of product. Say, if you're Corynebacterium glutamicum, you're producing glutamate. So we look at perturbations that would alter the way some step or all steps from glucose all the way to the amino acid, the glutamate, alter what we get, in terms of quantity, how much we get, that sort of thing. And then we analyze our data, both looking at quantitation under normal circumstances and quantitation of metabolites under these perturbations that we have defined. And we analyze the results of the perturbations of our network, and then we can make some recommendations. So if we look at a pathway, and again we're just taking this general pathway, we can look from A to I, and at steady state we assume the velocities are all equal to each other, so we can just group these individual reactions and give it one velocity because they're all equal. They're all going to be V1. And same from I to B. These two are, if we're in a steady state, we're talking about this is going to be this equal to this in terms of velocity. So we just group these and call these all V2. Same with I to C. We're grouping these two and calling it all V3 because again they're supposed to be equal to each other. But we group the linear reactions that we can group, and that simplifies the process of metabolic flux analysis tremendously. So we can assume that we can observe the pathway, its reactions, and its intermediates. And then once we have kind of gotten our bookkeeping down, we want to perturb the system. We want to generally, if we have three separate fluxes like we do here, we have J1, J2, and J3, we would need three separate individual perturbations required to show, well, how do these affect the overall A to B and C. So we would originate each perturbation in each branch. Now, in that way, we can test the flexibility of the branch point. Like maybe we can't alter the amount of C that is produced. So we'll know that we don't have much wiggle room in terms of flexibility from I to C, let's say. But we can perform our perturbations in a number of ways. So we can do it locally. We can look at one enzymatic reaction. We can perturb it, and here is shown by this green P here. We can perturb it in such a way that we change the specificity of an enzyme. Maybe we add an inhibitor to the system, an inhibitor to a certain enzyme, and that changes the flux. So that's going to give us a new velocity here. So it's designated V1 new, and that will affect the flux J1 potentially. And that could easily affect these two fluxes, J2 and J3, as well as the velocities V2 and V3, or maybe not. And we would find that out, at least in terms of the velocities. We could affect the metabolic network globally over the entire pathway. This could be done by adding more substrate. We could add 10 times the amount of substrate we'd added before, so substrates in vast excess. We could starve for a certain substrate, so they could run out at a certain point, or they could have to make do with 10 times less substrate, and that will perturb the whole system, and that will perturb both the J1 flux, J2, and J3 here. So you'll have new velocities for each of these different reaction groups. And we also talk about metabolic control analysis, which is a little bit different than flux analysis. Flux, we're measuring what is, and in metabolic control analysis, we're concerned more with how this comes to be. So we quantitate the degree of control exercised by each individual pathway. Is this velocity, this A to I velocity, is this a rate controlling step in the big scheme of things? We can ask questions like that. We can do that by grouping reactions the way we have done, and then look at group control coefficients. So we can eliminate, depending on what we're looking at, we can eliminate cross interactions. So you can imagine the A to B and C pathway as part of one big metabolic network, and there may be a lot of things coming into and going out of it. Maybe A is an intermediate, as it turns out, or maybe B is an intermediate. So, or even one of the unnamed intermediates is intermediate for someplace else, for some other reaction. So we can eliminate any cross interactions that aren't relevant. And maybe under the conditions you're operating, you don't produce an intermediate for a different pathway out of that. We can eliminate what we don't need. We can restrict the communication, and this term communication is the carbon flow through our branch point. So that's what we mean by communication. And then establish the degree of flux control that's exercised by these different pathways. So what we're doing is we're infusing the concepts of integration, quantitation, and relevance in the study of biological systems. And this becomes multidisciplinary just by the nature of how we're doing things. So obviously the biological system component, we've got biology involved, medicine as well, chemistry, because we're getting into the molecular level, biochemistry, organic chemistry. So we're basically talking about biochemical interactions here. Chemical engineering, we're looking at production of product. We're looking at how to make more product. Computer science, a lot of the big component here, as we predict what our perturbations are going to do. Electrical engineering, of course, we're hardwiring the system in a way, so we can take a look at it in that respect. And of course I mentioned medicine and a variety of others. So metabolic maps are very interesting. Here is a metabolic map. Very complex, lots of stuff going on. You can see the TCA cycle, the Krebs cycle right here. You can see a lot of stuff coming off of it, a lot of stuff feeding into it. And that's great. You've got a nice metabolic map showing what goes where. But they tell us little about these metabolic maps, tell us little about the flow of carbon. How much carbon is flowing into the TCA cycle? How much is flowing out of it at any given turn of the TCA cycle? We don't know about the flow of carbon or even any other elements through these pathways that are shown on this map. Just like a regular map, a road map, is going to show you what streets are in a given town, but they're not going to tell you how fast you're going to go when you get to from one point to another. So we have the map, but we need our flux analysis and we need our metabolic control analysis to really give us data on what perturbations and how perturbations affect the overall big picture, as well as the bunch of little pictures that you'll be creating looking at different pathways. And again, why does all this matter? Why go into this granularity, this amount of detail? Well, for one, I mean, we're talking about cellular and organismal health. We could be talking about nutrition. A lot of metabolic flux analysis is involved, looking at how nutrition affects our cells and other types of cells. And largely this was born out of a need for advancing biomanufacturing and biotechnology, where we're looking at inputs and then we're definitely looking at outputs as a product. We're looking at inputs and outputs in all cases, but in this case, in biotechnology's case, output is our product. And then the goal becomes maximizing production, whereas for cellular health, obviously the organism staying healthy, that is priority number one. Okay, so we won't go into this think, pair, share. I'll just tell you about it. So we have two organisms, Pyrrolula species and E. coli. Both organisms make a useful secondary compound, let's say. Which organism should be used to perform metabolic flux analysis and metabolic control analysis, and why? So we can think about these two species here, and why wouldn't we want to use either of them? Why wouldn't we want to use this Pyrrolula? Who cares? Had you even heard of Pyrrolula before I mentioned it just now? Well, yeah, you may not have ever heard of it, and there's a reason for that. The organism is not very well characterized. E. coli, on the other hand, is so well characterized that there are diverse, not only a lot of papers published on E. coli and what it does metabolically. But diverse reference books are devoted to, in this case, these black books here are talking about all the different pathways, metabolic pathways, that are present in E. coli. And there are a bunch of books that are published on E. coli, like how to manipulate E. coli, and different manipulations that have been done and will be done in E. coli. So there's a lot of material on E. coli. So you perform metabolic flux and metabolic control analysis on an organism. Your first thought is that you must have a well-characterized and well-understood system. And if I'm choosing between E. coli and Pyrrolula, well, I'm going to choose the E. coli. Pyrrolula would be a poor choice of organism since we don't know a whole lot about its metabolic control parameters. They're not well established. That's not saying they won't end up being well established in the future. At the current time, they're not very well established. So better to go with E. coli and using it as your microbial chassis, your choice, your organism, your biocatalyst organism, if you will, for metabolic engineering. And there could be a variety of driving forces behind metabolic engineering. In medicine, that's a key one. The creation of novel drug platforms, drug effectiveness. A lot of drugs are metabolized. In fact, most drugs are metabolized to some degree by our cells. So we would easily be able to use metabolic engineering to analyze how effective these drugs are. And health and nutrition, your overall health, your cells' overall health and nutrition, and also the effects of environmental insults. So not necessarily drug effectiveness, but toxin effectiveness. How effective is a toxin in killing cells, that sort of thing, or slowing down metabolism especially. And again, this is a good thing that we can use metabolic flux analysis for. For industry, the platforms are quite obvious, I think. Again, making products, biofuels, bioplastics, nutraceuticals, drug manufacturing, organic compounds or precursors, things like propane diol and things like that, can be made fermentatively as opposed to just straight standard synthetic chemically, as it were. So we're quantifying metabolic flux in cells. We're analyzing metabolic control strategies or network control strategies. And we end up using molecular biological techniques to implement certain modifications, making certain perturbations in our system of our biocatalyst organism. And all of these things are what make metabolic engineering what it is. They are the essence of metabolic engineering, as Stephanopoulos and others say it. So essentially, this is a quick introduction to metabolic engineering, the goals, what the procedures are, and there's a variety of goals. The goal, in a nutshell, is to establish better biomanufacturing processes, to make better biocatalysts, to better analyze how certain things affect our cells. Those are some goals. The procedure itself, how do we get there? Well, really 3 steps. Know the system. If we're talking E. coli, we're talking Saccharomyces cerevisiae, we're talking human cells. We know those systems for good reason. Perturb the system in some way, and then analyze the outputs. And then potentially go back and perturb the system again, and just keep doing that for a number of iterations until you get the results, or as close to the results that you would hope to get. So, many of you don't have the book yet, but when you do, or at some point, I suggest you begin by reading the first chapter, the Stephanopoulos et al. Book, pages 1 to 20. Kind of talks about what I just went over. Also, many of you already know about this, but those that don't, I want to do a muddiest point paper in class for every lecture that I give. So essentially, what is this? It's also called the minute paper. If you didn't understand something that I said, like of showing all these pathways, these fake pathways, got your eyes a little dizzy. Well, then write down a question that you have on a piece of paper and hand it to me before you leave the class. Or did you learn something? You can feel free to let me know about that. But again, you want to write it down on a piece of paper and hand it to me before you leave. And if you didn't understand something, we'll go over it in the next class. Obviously, we can't do that today because we're not in class anymore. But in general, this is what you want to do. And if you have questions about what we talked about in class today and in this little video, you can feel free to send me an email before Wednesday's class. So note that this muddiest point paper is not required. It's never required. It's there to help you. It's highly encouraged. So again, if you're not understanding something, please let me know so we're all on the same page, because it's going to be a requirement as you go forward to know what we're doing as we go along. So to begin to have a basis of network flux analysis, you already need to know about metabolic flux analysis, that sort of thing. Don't get left behind. Feel free to ask early and often about clarification on certain topics. Okay, thank you, and I will see you on Wednesday.